Hi, everyone. So my Keystone is also my biomedical engineering senior design project titled In-Silico Prototyping for an Intranasally Administered Agent for COVID-19 Prophylaxis and Treatment. So this project was done in a team. So all the data and the figures that you're going to see were created collaboratively. So as we all know, the coronavirus pandemic continues to place unprecedented pressure on global healthcare systems. And although recent vaccine efforts have been promising, it's possible that mass immunization will only be achieved in many low-income countries by 2024. So I think to combat the inequitable distribution of vaccines, there's real value to exploring alternate solutions to contain the spread of the disease, especially targeted to low-resource communities. So as a reminder, vaccines work as preventative measures. They mimic exposure to the virus to prepare your immune system for future infections. Another approach used by the company Fractal Therapeutics is to develop a combination of drugs that could work as early treatment right at the first site of the infection. So the first site of the infection of the virus is the nasopharynx. So if you remember getting tested last year, the nasal swab had to go all the way up to that region. So to deliver a drug combination to that region, we need to develop nasal sprays or inhalers that can be used by non-experts to administer the drug. And we probably can't just use over-the-counter nasal sprays because they're targeting different regions of the nasal cavity. And we also don't know if they will consistently deliver the required amount of the drug to the target site. And so this is where our project comes in. We are a small link in a large chain in a product development pipeline. We are identifying optimal properties of the drug formulation as well as the delivery method using a computational approach. So the results of our study are going to be used by other engineering teams in the future as recommendations to design nasal sprays treating COVID-19 or even other respiratory diseases. So I just want to go over our workflow for a bit. So we used models of the nasal cavity that were constructed from computed tomography or CT scans of 2 patients. And then we use software to simulate airflow in the nasal cavity. And essentially what the software does is it sets up and solves some really fundamental equations that describe how fluid behaves in a particular context. So lastly, we use the same software to simulate the release of our drug formulation through the nostrils in a way that can mimic a real nasal spray. So the goal of our project is to maximize the number of particles that get deposited at the nasopharynx. So this is really the workhorse of the whole project, but there are many parameters that we needed to set up in our framework to test. So I just want to highlight a few key ones. The first is looking at the airflow turbulence scheme. We can have either laminar flow or turbulent flow, which we also refer to as LES. A laminar model is really good for approximating a resting breathing rate or a low flow rate, and turbulent flow is usually seen at higher flow rates. So then when looking at the drug formulation itself, we wanted to vary particle diameter from 1 to 24 micrometers. So commercial devices usually have larger particle sizes, but smaller particles have a better chance of reaching all the way back to the nasopharynx just due to the physics of the flow. So for the delivery method itself, we started with looking at something called the current use protocol, which I will go over in the next slide. So essentially, you are told to hold the bottle upright and then lean forward a little. And this protocol results in an angle between the nasal spray and your nose that's around the ballpark of 67.5 degrees. And you're also told to insert the bottle 5 millimeters into the nostril. So we simulated the current use protocol for a single patient, and I will show you the results now. So for each nasal cavity, left or right, we simulated 2 flow rates. So you can see that the top series is 15 liters per minute, and the bottom series is 30 liters per minute. And so I just want to draw your attention to this zoomed out graph. So the peak here simulates a scenario where all the particles are 14 micrometers in diameter. And you can see that approximately 0.6% of them reach the nasopharynx. So that's indicated by the y-axis here. So I just want to say that the takeaway of this plot is that we're getting very low deposition, not really close to the amount you need for treating the virus. And so just for context, we need at least a few percentage points of deposition, if not more. So we try to go back and see if we can vary some parameters to improve these results. And so just intuitively looking back at the model, most of the particles look like they're going to get trapped in the initial regions of the nasal cavity and probably won't make it back to the nasopharynx. So we also saw this in our data. There was a lot of off-target deposition. So one way we wanted to try to fix that is to change the angle of the nasal spray. So that's what we did. We came up with a perturbed direction using a set of guidelines. So after drawing the axis of the nasal spray, we want the nasal spray axis to hit the nasopharynx, but we, as points 2 and 3 show, we want to minimize the axis hitting any off-target locations as much as possible. So essentially, we're trying to use this like line of sight method to target the region. And so we ran simulations with the perturbed direction and ended up getting much better results. So all 4 cases show that the perturbed direction of the nasal spray axis result in much higher deposition peaks at the nasopharynx in comparison to current use. So in fact, in some cases, like in plot B, we even saw up to 100-fold increase in deposition just from varying the nasal spray axis. So this is all just for one patient. We wanted to see if we could observe a similar trend in a second patient. So we did the same procedure, finding a perturbed direction. And here are our results. And in most cases, we can see again that the orange line, which is the perturbed direction, does do better than current use. So one thing to consider in this case is that it's pretty difficult to precisely orient a nasal spray bottle along this like exact angle. So in clinical settings, you probably will see a lot more variation in the angle. And so we wanted to test if the perturbed direction was tolerant to slight deviations. And so what we did is identify 5 additional perturbed directions, starting from the original one, which is in dark blue. And we tested deposition for each of these different cases. So I'm going to show you the plots now, and there are a lot, but essentially the takeaway is that there's high correlation between the perturbed directions in each plot. So this essentially tells us that the perturbed direction is tolerant to small deviations and that it is pretty robust to these changes. So it's promising if we want to adapt it to a clinical protocol. So now one other thing that I want to mention here is that we have only used 2 models for our entire analysis, but noses vary really significantly between people across the population, as you all probably know. So however, if we want to extend our analysis, we are really limited by the fact that CT data, computed tomography data, is really not available, readily accessible across the world, especially from low resource settings. So to address this problem, we wanted to see if we could gather estimates of how specific features of nasal anatomy change with different demographics, such as age, race, gender, and then eventually use a statistical modeling approach to make these synthetic or artificial nasal models. And then we would eventually feed these synthetic nasal models back into our workflow to see, to test particle deposition for a larger population. So we started working towards this goal through a really extensive literature review. And we profiled journals ranging from disciplines like biological anthropology to rhinoplasty journals and even modern computational fluid dynamics studies. And so we identified just a few candidate anatomic features. And so I just want to highlight cross-sectional area of the nasal valve, which is marked in this figure. And this is one of the narrowest regions of the nasal cavity and contributes a lot to the resistance to flow. So this is an example of the kind of statistics we found. These are ranges for the cross-sectional area across 3 different groups of race. And essentially we have tons more data sets like this identified. So we really hope that this analysis has laid the groundwork for generating a really inclusive and globally representative set of synthetic nasal models. So just in summary, the study shows that it is possible to have intranasal delivery of drug combinations. And we identify both the optimal particle size ranges as well as the optimal spray axis for the nasal spray. And then the second thing is that we've started outlining a workflow for future groups to expand the diversity of nasal geometry models such that we can characterize intranasal delivery for a larger population. So I also want to acknowledge a lot of people who contributed to this project. Firstly, my wonderful senior design team, Phoebe Atto and Noge Ka, for being the best. And also Yu Ying Lau, who is a recent graduate master's student who also did a lot of work for this project. And of course, our fantastic advisors, Diane, Arijit, and Shoikat, for guiding us through the whole thing. And yeah, I can take questions now.